parameters used in expressing solute concentration, there are two parameters, that is molarity and osmolarity. So molarity is the number of molecules per liter of solution, while for osmolarity, it is the total number of particles per liter of solution. Sounds the same. So how to differentiate between these two? So some of the molecules, they dissociate in solution. For example, sodium chloride. Sodium chloride can dissociate in solution to produce two particles, which is sodium ion and chloride ion. So in this case, the number of molecule is 1, while the number of particles is 2. So number of molecules does not always equal to number of particles. So for the unit used for both parameters, the unit used for molarity is mole per liter, or known as molar. The unit for osmolarity is osmol per liter, also known as osmolar. So let us compare two examples. Glucose. Glucose does not dissociate in solution, so one glucose molecule equals to one particle. So 1 molar of glucose solution will only have the concentration of 1 osmolar. While for sodium chloride molecule produces 2 particles. So 1 molar of sodium chloride solution will have the concentration of 2 osmolar. So let's have a small test. So what is the osmolarity for the solution containing 1 molar of sodium chloride plus 1 molar of glucose? So let's see. 1 molar of sodium chloride equals to 2 osmolar, while 1 molar of glucose equals to 1 osmolar. So total up, we have 3 osmolar.
Is the net movement of water molecule down the concentration gradient across a semi-permeable membrane until an equilibrium is achieved, which means that water molecule will actually move from a diluted area to a more concentrated area. Secondly, we are going to learn about osmotic pressure. What is meant by osmotic pressure? It is actually the pressure applied by the water molecule to prevent the water molecule from entering the solution. And it is measured by two units, which is atmosphere ATM and also millimeter mercury mmHg. When the water concentration is higher, we know that the number of particles equivalent with osmolarity is lower. When the water concentration is higher, the osmotic pressure is also lower. So in short, we can say that in the diluted area, when the water concentration is higher, the solute concentration, osmolarity and osmotic pressure is lower. Whereas in the concentrated solution, when the water concentration is lower, the solute concentration, osmolarity and osmotic pressure is higher. Lastly, we are going to learn about extracellular osmolarity and also the cell volume. For example, the concentration of potassium is higher in ICF, which is intracellular fluid, compared to the extracellular fluid. So when potassium molecule move out of the cell, it is actually pumped in again by the sodium-potassium ATPase pump. For the sodium molecule, it has a higher concentration in the ECF compared to ICF. So when it enters the cell by ion channel or by secondary active transport, it is actually pumped out by sodium or potassium ATPase pump. For sodium chloride, it has a higher concentration for chloride solution. For the chloride solution, it has a higher concentration in the ECF compared to ICF. So when it enters the cell, it is moved out again by the electric impulsion. So ECF osmolarity is equal to ICF osmolarity, which it is 300 milliosmolar.
Tonicity and osmolarity. Tonicity can be divided into three, which are isotonic, hypotonic, and hypertonic. Meanwhile, osmolarity can also be divided into three, which are isoosmotic, hypoosmotic, and hyperosmotic. Isotonic. Isotonic can be defined as a solution containing 300 mol per liter of non-penetrating solutes regardless of the membrane-penetrating solutes that may be present. If a cell is put in an isotonic solution, the cell will undergo no change. Next is hypotonic. Hypotonic can be defined as a solution containing less than 300 mol per liter of non-penetrating solutes, regardless of the membrane-penetrating solutes that may be present. If a cell is put in a hypotonic solution, the cell will swell and undergo hemolysis. Then lastly, for tonicity is hypertonic. Hypertonic is a solution containing greater than 300 mol per liter of non-penetrating solutes, regardless of the membrane-penetrating solutes that may be present. If a cell is put in a hypertonic solution, for example, for 450 mol of sodium chloride, the cell will shrink. For osmolarity, isoosmotic can be defined as a solution of 300 mol per liter of solutes, regardless of the composition of non-penetrating and membrane-penetrating solutes. Next is hyperosmotic. Hyperosmotic is a solution containing greater than 300 mol per liter of solutes, regardless of the composition of membrane-penetrating and non-penetrating solutes. Lastly, it is hypoosmotic. Hypoosmotic is a solution containing less than 300 mol per liter of solutes, regardless of the composition of membrane-penetrating and non-penetrating solutes.
We use a few examples to clarify the basic concept behind tonicity and osmolarity. In example 1, we have 150 milliosmolar of sodium chloride as non-penetrating solute and 150 milliosmolar of urea as penetrating solute inside ECF. As for ICF, we have 300 milliosmolar of non-penetrating solute. ECF is isoosmotic to ICF. This is because the total osmolarity in ECF is 300 milliosmolar and is the same compared to ICF. ECF is hypotonic compared to ICF. This is because the concentration of non-penetrating solute inside ECF is lower than that of ICF. Therefore, water will move in by osmosis from ECF to ICF and causes an increase in cell volume. In example 2, we have 500 milliosmolar of sodium chloride and 100 milliosmolar of urea inside ECF. ECF is hyperosmotic to ICF. This is because the total osmolarity of ECF is 600 and is higher compared to ICF. ECF is hypertonic compared to ICF. This is because the concentration of non-penetrating solute is higher inside ECF. Water will move out from ICF to ECF and causes an decrease in cell volume. In example 3, we have 300 milliosmolar of urea inside ECF and 300 milliosmolar of sodium chloride inside ECF as well. We have 300 milliosmolar of non-penetrating solute inside ICF. ECF is hyperosmotic to ICF. This is because the total osmolarity inside ECF is 600 milliosmolar. ECF is isotonic compared to ICF. This is because the concentration of non-penetrating solute inside ECF and ICF is the same. So the cell remains unchanged.